Thank you very much for the introduction, and thanks to the committee for providing me the opportunity to speak today and discuss my data with you. For those of you who are unfamiliar with this field, I'd like to give you a brief introduction. So tumor necrotic factor, otherwise known as TNF, is a cell signaling protein, and we know that it's involved in mediating cell death and inflammation in various diseases and cancer. It was, the story really started about 100 years ago when a doctor by the name of William Coley, he actually gave tumor patients some extracts from bacteria, injected them, and then he realized that not only did it regress the tumor, but it actually caused a huge systemic reaction. And then years later, Dr. Lloyd Old, who was then the father of modern immunology, in 1975, he actually identified the same molecule in macrophages, supernatant of endotoxin-treated macrophages. And then his colleague Carswell, what he did is injected mice with a BCG endotoxin, and he realized that actually the mice produce serum factor that causes hemorrhagic necrosis of the tumor. So from this time onwards, it was thought that maybe TNF has some sort of part to play in tumor, in cancer research. So how does TNF exert its biological responses? Well, it binds to two cell surface receptors. These receptors show sequence homology on the extracellular matrix, but they're unrelated in the intracellular domain. They they don't have an intrinsic enzyme activity within the cytoplasm. So the way the signal cascade occurs is through interaction with adapter proteins within the cytoplasm. And depends on what the trigger, they can actually cause apoptosis, proliferative genes, inflammation, or survival. But then the thing is they can actually trigger individual molecules, or they can trigger the same molecules, and therefore they show both shared and non-identical signaling pathway. So why are we interested in RCC, renal cell carcinoma? Well, 2% of these represent adult cancers worldwide, and of the 2%, 90% of them are of kidney cancers. And out of those 90, 80% of the clear cell type. Now, these cancers are very important in a way that they've drawn a lot of attention because they show very poor prognosis. They're resistant to treatment with the radio and chemotherapy, and they originate from the tubular cells within the kidney. They're known to secrete TNF, and I've myself reported this in 2010. So that shows that TNF may play a role in the initiation and progression of the RCC, mainly the ccRCC. So a few years ago, in 2010, we set out to actually determine whether these receptors are expressed and whether they actually function in the ccRCC. So what we've done is on the left is the H&E, just showing the morphology. I term here a normal kidney as non-tumor, adjacent non-tumor zone, and obviously the different grades of RCC. There are normally 4 grades, but I only show 3 here. And on the right-hand panel is immunofluorescence that we've labeled with anti-TNF-R1 and R2. And what we show that in normal kidney or non-pathological kidney, R1 is constitutively expressed in the glomerular and vascular endothelial cells, while R1 is rarely found in normal kidney, and if so, very in maybe infiltrating cells. Whereas it's opposite in the RCC, whereas you have upregulation of TNF-R2 and mainly localized to the tumor cells, and as well as infiltrating cells. And at this point in 2010, we thought that actually R2 is expressed, these infiltrating cells are actually mononuclear cells. R1, on the other hand, is only found in the vessels and in some of the infiltrating cells. So then my interest was, okay, well, that's actually an observation, but actually what we need is to look at to gain insight into the tumor and find out what exactly do these receptors do. So what I did is I've developed myself an organ culture system in our laboratory, whereby I take pieces of tissue, less than 1 millimeter in diameter, or rather, yes, in diameter. We place them in culture, and then what I did is through our collaboration, my collaborator in Belgium, we altered a bit of the wild-type TNF sequence so that we can actually substitute the amino acid to bind to either TNF receptor 1 or TNF receptor 2. Thereby, we can delineate which of the receptor is signaling. So we can maintain this tissue in culture, prime them with this mutin or the wild-type TNF for 3 to up to 18 hours. We then harvest the tissue, fix it, and stain it. And what we find here that when you ligate TNF-R1, you actually induce cell death of these cells within the tumor. And when we did co-staining, some of these were tumor cells. And can I use the pointer?
Yes.
It's pressing, you know?
It's right here.
Yeah. So we use tunnel staining to detect apoptosis versus the caspase-3, and we find that wild type triggers cell death, and R1, which is R1 selective mutant, triggers cell death as well, but R2 doesn't, and this can be shown on the right with a histogram. And then the opposite happens when you selectively ligate TNF-R2. You actually induce cell proliferation, and more so with the wild type in R2 as opposed to the R1. And again, it's significantly, there's a significant difference, more so with the wild type than the R2. So then the question became is, what are the targets of TNF in RCC? So we know that we saw that those TNF-R2 expression in some mononuclear cells. So there's a lot of literature now coming out that stem cells are actually that are present in RCC, and we know that they are actually form a bulk of the tumor. They are tumor-initiating cells and have limited capacity for self-renewal. We know that they can generate any cell type. They're quiescent, slow cycling, and divide very infrequently. Again, they're very resistant to chemotherapy. That most of the chemotherapy targets cycling cells. So therefore, that's why we get recurrence of tumors. So now we know that maybe these cancer stem cells could be, the sensitivity could be enhanced by inducing them to divide. So this is what actually leads me to my hypothesis, is to test whether cancer stem cells, that if we drive them to proliferation, can we basically, by ligating R2, can we render them more susceptible to cytotoxic agents? So what we did here, we first again looked at normal versus tumor. We used CD133 as a marker, as it's been proposed as one of the stem cell markers. And what we find here is RCC has more CD133 expression, or expressing cells, and they actually increase with tumor grade, more so as this tumor becomes more severe. And then we know that they're actually stem cells because they express some of the stem cell transcription factors. And here I just show OCT4 as one of them. And then the other question becomes, well, can TNF-R2 has any effect on this CD133 in tumor? So what we did, again, in organ culture, I took the tissue, treated it with R2, R1, or the wild type. And again, I see that when you selectively ligate TNF-R2, you increase these numbers of cells that express CD133. And interestingly, that these cells actually correlate to an increased expression of the R2, and they are the ones that show proliferation by phosphohistone staining, which is one of the markers for proliferation. So then the other question that was, can we actually isolate these stem cells from their kidney tumor? And so what we did is we used this is a technique that's out there. It's a MAX technique. It's magnetic activated cell sorting. So what you do is you cut pieces of tissue, you digest it using this disassociator, and you pass it through. You label the cells with a magnetic beads that have CD133, and then you incubate them, pass them through a magnetic column, and you get positive selection of CD133 positive cells. So, so these are actually stem cells that come from the tumor. So what we did here is a phase, just to show you a phase contrast image showing the morphology of the cells. On the right is immunofluorescent with CD133 marker, and they're all staining. They show stem cell markers or stemness markers, but they don't actually show any differentiation markers. So from this time onwards, we know that actually we are dealing with stem cells. And then we wanted to find out, do these actually express TNF and the receptors, but in culture? So again, R2 is expressed at 3 hours and 6 hours. Hardly R1 or wild type is detected, but actually they increase in time, especially TNF-R2. This is just a representative image of the results. On the right is the relative mRNA expression with the qRT-PCR. Again, that's the treatment on the x-axis. When you treat with wild type, R1 or R2 ligation, you increase TNF message, while the opposite happens. When you ligate R1, you increase TNF R1, and ligation of R2 increases the TNF R2. So then we wanted to find out what happens then if you prime these cells with an R1 mutant, or the wild type of the R2. And what we find is, again, using tunnel staining as a detection for apoptosis, wild type and R1 increases death of these cells, and more so at 18 hours than 3 hours. And some of these cells that are dying are actually showing expression of TNF-R1. The opposite happens when you ligate with R2. You actually increase cell proliferation of these cells, here in pink, and we use phosphohistone as a marker for cell proliferation. And again, these cells express TNF-R2. So here, the flow cytometry results were concordant with our immunohistochemistry, but in this instance, we use proliferative cell nuclear antigen as a marker for cell proliferation. And again, you get a shift on the right with the R2 mutant and the wild type, but not the R1. So then the next question becomes, well, okay, so we know that the R2 receptor can actually be transcribed. It can be induced by both the protein and the mRNA. So the next question really is, can we actually prime these stem cells with the R2 to drive them to cell differentiation and towards the epithelial phenotype? And the reason we are interested in epithelial phenotype is because RCC is actually a tubular epithelial cell tumor. So we were interested, so we used a 3D matrigel. And again, on the left are the treatments and the days in culture. And you see that actually there's spheroid colony formation in day 2. And there's an outburst of cells, day 4, day 7. In R2, they start forming network-like structures. And then in day 14, you actually get like a tubular-like formation. And interestingly, you get simultaneous loss, actually, of the stem cell markers or transcription factors, but you don't actually lose OCT4 and Lin28. And this wasn't actually very surprising to us, because we have previously reported this in a model of myocardial infarction, which I'm not going to go through, but I'm happy to discuss with anyone who's interested after. And then interestingly, you start getting induction of differentiation marker, more so cytokeratin, which is a epithelial phenotype marker, and APCAM, that epithelial adhesion molecule. And the thing is, you start gaining TNF-R2 in these tubules, and TNF, not so much of R1, hardly there. So then, what really clinically, maybe that's relevant, but really for clinical trials, I think what would be interesting is, now the thing is, we know that stem cells are resistant to chemotherapy. So can we actually drive them to proliferation, to make them more sensitive to anti-cancer agent? So what we did here is we took, I'm just showing a simple data really, is to take the cells, the stem cell that have been isolated from the tumor, treat them with we use cyclophosphamide here, by the way, because it targets the cell cycle. So what we did is, and sorry, this is actually used in the clinic. So we wanted to try it in the laboratory. And what we did is we took the cells, either untreated in media only, treated them with R2, or the cyclophosphamide on its own, or drive them to cell cycle first, and then treat them with cyclophosphamide. That actually, the lower one was what we were interested in. So this is actually just flow cytometry data, really, Hirsch just to show the nucleus pattern, and an axin for apoptotic and necrotic PI. And you see that when you drive them to proliferation by R2 first, and then treat them with cyclophosphamide, you drive them to proliferation, kill them, and they actually show more effects of the chemo drug than when actually you use the chemo drug on its own. So really, in summary, we know now that our stem cells, cancer stem cells in CCR can be studied in situ and in vivo. We know that when you ligate TNFR2, you can increase proliferation of R2, its transcription of R2 itself. You can promote these cells to enter cell cycle and differentiate into tubular structures. We also know that when you ligate TNFR2, you can render them highly sensitive to an anti-cancer drug. And overall, really, our results show that TNF may provide an important signal for differentiation of CCR6 cancer stem cell, and that could maybe be exploited in the clinic, really, for killing and eradicating tumors. Thank you for your attention, and I'm happy to take any questions. Or just to say, this is Cambridge, by the way, in summer. Everyone, this is like one day in the whole year. And that's the famous King College. Thank you.
I have a couple questions. Excellent work.
Thank you.
Excellent
thank you.
Presentation.
Thank you.
And one is you showed that the TNF receptor 1 is selectively expressed in endothelial cells, and selectively activates TNF receptor 1 cause cell death.
That's right.
Is the tumor cell dying, or is other cell type? It didn't show.
Well, predominantly were tumor cells.
If it's tumor cells,
yeah.
Have you thought about why you activate TNF receptor 1 on the endothelial cell surface and cause tumor cell death?
Well, I think that is something we can do, but that's not the direction we wanted to take. So we were actually... No, that's a very good point. I think we should, because, I mean, there's a lot of angiogenesis in RCC.
Because that's, you can use tumor necrosis factor to actually kill the tumor cells through endothelial cells.
Yeah, you can, but our target was the stem cells, because they are the ones that keep the tumor going, you see?
Yes. And the second question is about the cancer stem cells.
Mm-hmm.
You showed that actually in the RCC cancer stem cells, there are expression of 4 so-called pluripotent stem cell transcription factors.
Right.
LIN28,
OCT4.
NANOG, OCT4,
and NANOG.
Yes, SOX2.
Right.
And we know that fast expression of these 4 factors can induce pluripotency.
Right.
So convert that to a pluripotent stem cells.
Right.
How about, can you culture your so-called stem cells in culture infinitively as like a iPS
a bit
cells,
like iPS, yeah.
They'll die eventually?
No, I think you can, but I don't think that would be that's not our direction, but maybe it's possible. But the problem then becomes is, you see, when you deal with tissue, it's totally different to dealing with a single...
cells. Right. You isolate cells by the magnetic sorting.
That's right.
Okay. How about the behavior of these cells in culture?
No different to any other cells that we've used in the MEC system.
They eventually die, right?
You mean once you isolate them?
Isolate and put in culture.
No, they don't. I think you lose a lot of them, but they don't die.
They are differentiated, or
you have to drive them to differentiation. In my drive, yeah.
Okay.
Nice question. Thank you.
Other questions?
We have one more question right here, sir.
That sounds counterintuitive that you are growing the cancer stem cells and trying to kill them then. So I think the clinical question is how efficient you are. Because if
you mean in killing them?
In killing them, exactly. Because if you kill just a percentage, you end up with the same amount of cancer stem cells, which is not good, of course.
Well, that's a valid point, but I think at this point we need to test a bit more drugs to be able to say whether if we are able to tune to which drug we cannot kill all of them. I mean, the thing is, the whole point of the experiment really is because we know they're chemo-resistant. And we know that in clinical, that's why the drugs fail in the clinic. And the question then becomes, is instead of using TNFR1 to just directly kill them, because then it's probably there'll be an adverse, probably inflammatory reaction, then maybe the best way to do is go indirectly and say, well, can we actually sensitize them so that they can respond to the anti-cancer agents?
Then maybe just a quick question.
Yeah.
You don't have to answer it.
No, it's fine.
How much reversing can you create by removing the receptor activation? Can you reverse them?
Well, okay, so I didn't show the data here, but as I said previously, I actually published a paper on how important when R2 is. For example, when we take a mouse and we knock down TNFR2, you have a reverse effect. You don't have lots of stem cells, and you don't have any effect at all. So there is a way where you can maybe reverse it by manipulation of the receptors, but that's something we've never done in this model. So we don't. I think there's a lot we can do.
Yeah. Thank you very much.
Yeah, thank you.
So thank you.
Thank you very much. Thank you.